Hello and welcome. It's now 4 p.m. and we are going to start this webinar where I will talk about RNA-seq and single-cell data analysis. And I'm Ann Holmberg at Qlucore. I will be your guide for the next 45 minutes or so. And you are muted, so please use the chat if you have questions. I saw that some of you used the Q&A. I'm not monitoring that, so please use the chat. And I will send out the slides after this webinar to all of you. And then I will stop my video because I'm using another screen. Okay, and Qlucore Omics Explorer, as I guess most of you know, it's a very fast and easy to use tool to analyze and explore data without being a statistical expert. And that being said, we also have statistical experts using this software, but the majority of our users are bench scientists and medical doctors. And to give you a quick overview, what the software can do is we can visualize and explore data, check quality, look for outliers, find structures in the data, and generate new hypotheses. And this, we will have a closer look at this when we look at the single cell data set. We will try to identify subpopulations of the cells. And when you have a hypothesis, you can perform statistical analysis. So we have a couple of built-in methods. We have a couple of methods that are available via our interface to R. Here you can also plug in your own R-scripted methods, and you can easily export your results as lists, reports, and plots. And once you have found a list of interesting genes, proteins, metabolites, you can, for instance, use the built-in gene set enrichment analysis to perform pathway analysis. And if you have a data set with very strong and clear signals, you can use that to build a classifier. Using the built-in machine learning algorithms, KNN, support vector machines, random trees, or XGBoost, and then you can use the classifier for prediction of the same groups in a new data set. And then we have one separate module in this data set, which is the NGS module, which contains a genome browser. So if you're interested in having a closer look at your genes, look at the actual sequence, you can visualize that in the genome browser, look for variants. If you're working with gene fusions, you can visualize them here as well, and also show them in circular plots. And in the new version, we just introduced also support for ChIP-seq and ATAC-seq data here in the genome browser. And you can work with that in parallel with your normal expression analysis, so you can have that in parallel windows in the same session. Okay, and we support a lot of different data types. Today we are going to focus on RNA-seq data as well as single-cell RNA-seq data, but we also support a lot of other data types such as DNA methylation, proteomics, metabolomics, and so on. And we support a couple of file formats, and many of these are RNA-seq file formats, so I will show you a lot of them now during today's demo. We also support cell files for mathematics, Thermo Fisher, Agilent files. You can download data from Geo, which I will show you today, as well as download of TCGA and 10x Genomics data, also included in today's demo. We can load data via the wizard, which I will also show. And here are file formats supported by the NGS module. So we basically support any multivariate data with this software. So the agenda for today is that I will show you a lot of different ways to import RNA-seq data. So I will focus on how to get the data into the software, not so much on how to analyze that. I will give you some brief examples only. I will also show you how to add annotations to a data set, because when you load BAM files, for instance, you don't get any annotations with that, so you need to add that in a separate process. We're going to have a look at how to download data from Geo, and we will end the session with analysis of a single data set from 10x Genomics. So let's get started. So I will start to show you how you can import aligned BAM files. So here we need to have the reference genome as a GTF file that is the same that was used during the alignment. So it's important that you have a matching GTF file here. And let's see how that works. So I open up the software here. Here we are. Here we are. And then I go here, File, Open BAM Files. And here you see, here we have the GTF file. It doesn't remember the one I used last time, so let's go and find it. Here I have the one that I want to use. Here I can do some pre-filtration if I want to or not. I decide which of the 3 supported normalization methods that I want to use, and here I have the possibility to also work with stranded data if the protocol that was used was for stranded DNA. And then I select the files. So I go over here, find my files. We are, and here I have a couple of BAM files, and you see they are very small. They are 10 times smaller than what you get when you do whole genome sequencing. But that's good when you want to demo. So I pick all 4 of them like that. Now I have them here, and then I just press OK. And now the process starts. So I start by counting them. Finding out how many reads do I have per gene, creating a matrix, and then we do normalization according to the normalization method that you chose, and we end with a log transformation of the data. And here is the QC report, some information. Here we have the base position quality. Since these are quite small BAM files, this is just a straight line. Normally you get a declining curve here. And here we have the Spearman and the Pearson correlation. And you can export this information and also save the report if you want to. And when I close this down, I have my data here in a PCA plot. As you know, any data that you load will always start in a PCA plot. So here I have my 4 samples. If I want to have a look at the normalized data, I have it here. So here are the normalized and log transformed values, like that. And if I look over here, I see that I don't have any sample annotations. So in order to be able to analyze this, I need to add sample annotations. And I will show you that a little bit later. But after normalizing data or reading it in any way, you probably want to do a save as a GE data file. So next time you want to load this data, you don't have to go through the full process. You can just open the GE data file, which then has the normalized values in it. So that's always a good thing to do. Save your data set as a GE data file, and then you can quickly load it the next time you want to work with this data set. Okay, then we close this down and we proceed. Now we're going to have a look at how we can import a raw count matrix. So sometimes this is the format that you get your data as a matrix of your raw counts. So let's see how that works. So now I want to import this using the wizard. So then I go here, File, Open with Wizard. I go and find the file. Here it is. So it's a TXT file. That opens up the wizard. And now I can see it here. So this looks like a normal text file. I have the sample information here. I have the variable information here, and I have the measured data here, the actual counts. What you can see is different from what you normally maybe are used to is that here we have a column for the feature length, the gene length. And we need that information when we do the normalization, because that's one of the factors that we take into account when we do the normalization. So the longer the gene, the more the counts you expect for that gene. So this is a factor that we are using. And then we answer the questions in the wizard. The first question is this a raw count matrix? Yes, it is. That's important. Next, normalization mode. So I have the same 3 methods here to choose from. Next, input mode for feature length. Is it already in my file as a variable annotation, or is it in an external file? It's in my data set here, so that's correct. Next, select separator. It's tab separated. That's correct. The variables, the rows are variables. That's also correct. And then select the top left cell of the data matrix. That's my first measured data point. Next, select the top right cell over here. So now I've marked what is the data. Next, and then click on variable annotation header cells. And I also need to click on include this gene length column. Next, and then I mark the sample annotations, and then I press next or finish. And that should, sorry, I also need to select the feature annotation. So I need to tell the wizard where do I have the gene length. Now I can press finish. Here we are. Here is data. Here we have 12 samples and 50 variables. We have the normalized data set here. And in this data set, we actually have the annotations already in here. So I can select one of the annotations. I just changed that color by double clicking. Like that. Then I can perform maybe an F-test, a multi-group comparison on treatment, find the best discriminating variables, maybe open up a synchronized plot, make that a heat map, go to the View tab, press Auto, hierarchical clustering, color according to treatment, maybe change the symbol to. So I see I have a couple of questions. I will answer them in a little while. Like that. So here we have the results. So we see that we have a couple of genes here that are differently expressed between the different groups. So this is very straightforward. So in this case, you actually had everything, all the annotations in here. And again, you want to save this as a G-data set, and because then next time you don't need to perform the normalization. So let's have a look at the questions. So I got 2 questions here, I think. So the first one is, what are the differences for these 3 different normalization methods? Yeah, I think you need to Google that, because there are small differences. They basically, I would say, do the same thing. They account for the gene length and the read depth. For TMM, which was the other question, what does that mean? And yeah, that was my last example. The TMM factor, that's the amount of RNA in the sample. So that's also a normalization factor. So we take into account how much actual RNA is there in the sample. So that is calculated as part of the normalization and then used as a factor in the normalization. And another question, how does such a separate file for feature length information look like? What information should it contain? So let me get back to you on that. My guess is, and I think that's correct, that it's a very simple text file with the first column including the unique variable IDs, and then the second column including the gene length. But I will double check and come back with the, on that topic to you, Hanna, or maybe even send it out when I send out the material to all of you. So let me see. Let me just make a note of that. Format for gene length. Yes. Good. And then the next question is, can you export the plots as SVG or PDF files? Yes. So if I right-click in the plot, select Export Image, you see here you have so the heat map cannot be exported in SVG, but many of the other plots can, but PDF definitely. And the next question, is it possible to include the normalization method for DSEC2? So far we don't support DSEC2, but that's on the list to do. So we hope to include that in the upcoming version. So far, no. So if you want to use that, then you can pre-normalize your data and just read in the pre-normalized data set through the wizard. Okay, so that was all the questions for now. So I close down the chat and continue. Or was there one more? Okay, here. After importing the BAM file, is it possible to analyze abnormal splicing event, allele balance alterations? Well, so in the NGS module, you can visualize your the different gene splicings as well. So I guess you can do it there. I'm not sure about this specific question, allele balance alterations. But you are very welcome to have a look at the NGS module, which is also active in your trial license, and yeah, see if this is supported there. I can check this as well. So let me make balance alterations. Andrea. Okay, great. Okay, then we continue. And one more question. In the analysis of BAM files, can you limit the analysis to a particular gene type, inRNA, or coding? No. Coding gene? No, I think we will pick everything that's in there. Yeah, we will grab everything in there. So then the BAM file must be, so I don't think that you can put that filter on it during the normalization. Okay, now we continue. We can also import data from 10x Genomics Cell Ranger pipeline. And here we use a predefined template for import of 10x Genomics Cell Ranger data. And these are Python scripts. So let's see how that works. So I close down this data set. And then we open the template browser here. And here we have the 10x Genomics Assistant. So here you can see that here we can load data that is processed by the 10x Genomics Cell Ranger Pipelines count or AGR. And so the directory that we use must contain at least 3 files with these names. And they will create it if these pipelines are used, count or AGR. So let's find the library here, the path. So here we are, and you can see that we have these 3 file types here. So I just copy the path and I paste it in here now, and then I execute. And this takes a little while. So while we're waiting for this to process, it's quite a big data set. I'm going to talk a little bit about our the templates. So in here you can see we have the 10x Genomics Assistant template. We also have other ones, one for download, FTP download and processing. You can also perform internal workflows in the software. So here we have one where we do an F-test and pull up some plots. Here we have one where we do t-test and fold change and pull up some other plots. And here we have one for downloading TCGA data, which I will show you next. And all of this is done in Python. So if you have access and knowledge of Python, you can perform your own, build your own templates and plug in here. And there is a very extensive information about how to do that in the reference manual that you will find in the help section. So here you can really automate your analysis if you would like that by building templates. Okay, and now we are almost done. So then we will load a data set that's containing more than 1,000 samples and 30,000 variables. Yeah, so now I hope that this is done. Yes, now we are there. Yes, and we got a little error message that we have built it for version bigger or equal than 3, but this was actually generated from an earlier version, but it actually works anyway. So here we are. So here is now our data set with 1,100 samples, as you can see, and 32,000 genes. And if we look at the data here in the table view, you can see that there are a lot of zeros in here. So we probably want to do some pre-filtration. And in the data tab here, you have that. So we can say that we would like to remove variables that have a value less than something low. In maybe, for instance, 70% of the samples, that will get rid of the genes that have basically mostly zeros. Then we press OK. And then you see that then I filtered away 18,000 of the 28,000 of the genes. I only have 4,000 left. I can also see that the data is not log transformed. So that's generally a good thing to do, to log transform. So if I have zeros in my data set, I need to threshold to something low, and then I can log transform my data like that. And then I have my PCA plot here, and you can see that it's still hard to see anything. So we might want to also add a step where we do some variance or standard deviation filtration to be able to focus on the most interesting genes. So that opens up the data set a bit. So we will have a further look at this when we look at the single cell data set later on. But these are some tools that you have when you work with this type of data, where you really need to filter down and pre-process in order to take away noise and see something. And this was, of course, also a data set with no annotations, only the unique sample ID. So here we also need to add annotations. And again, we probably want to do a save as because we don't want to process the data next time we open this data set. We want to save it as a GE data file. Okay. And the templates works if you are working on if you are on a Mac, then you need to have version 10.15 or later in order for the templates to work. Then we can also download data from TCGA, and here again we have a predefined Qlucore template for that. So let's have a look at that. So we close this data set, and now we go here to the template browser again, and here you see we have a TCGA template. And here we are. And here we have data from prepared data from the Genome Data Analysis Center at Broad Institute, and we have prepared 27 out of 38 cohorts of different cancer types, as you can see. So I pick the cohort that I want to work with. I pick this one, ESCA, and I just type that in here. And then I can decide if I want the data to be normalized or not. So yes or no. And then I just press execute. And then we will download and process this data, which is, yeah, very fast. So that actually produces a data file as well, as you could see. Okay, here is the data. So 196 samples and 20,000 genes. And it's already colored according to histological type. So if we have a look here, we can see that with this data set, there are also a lot of annotations. So we have the histological type here. If we want to change the color, as you can notice, the color scheme has changed in this new version. So you might want to sometimes change the colors manually. And there is also an option to go back to the old color scheme if you like that better. Okay, and here we can do, of course, a statistical test directly. So I open the statistical dialog here. Maybe I want to do a t-test on histological type. So if I filter down to a Q value of around 0.01, I might also want to filter by fold change, and we see that I get very strange fold change values, and that is because this data is not log transformed. So let's do that. So in order for the fold change filtering to work, you need to log transform your data, because the calculation that we do assumes that the data is log transformed. So now I log transform the data, and now I get reasonable fold change values. So let's say maybe I want to filter to 3 or so. Let's go back to the PCA plot, and I want to filter down a bit further in order for the heat map to also being able to cluster the heat map on the variable side. So the default limit for that is 1000. I ramped it up a bit to 2000. So now we found 1700 genes, and we see we have quite a good separation between the two histological types, some exceptions. And again, of course, we can pull up a heat map and show these genes in a heat map. So again, I want to do hierarchical clustering, and I probably want to color on histological type like that. And here we are. Yeah, there was the network. So it becomes a bit slower when you go over 1000, as you can see. But here we see we have very clear signals here. Okay, so there you have a gold mine of interesting cancer data to dig into if you're interested. Then we continue. we also support data from the salmon pipeline, so quant.sf files that can come from salmon, and is also the output format used by Illumina-owned platforms. So let's see how that works. Let's just close this down. And then we open quant.sf files and we select the folder. Here we have that. Okay, so here's the folder of my samples. So I just pick, okay, select folder. Here are my 4 samples. Press OK. And again, they open up in the PCA plot, my 4 samples. If I want to look here in the table view, I see that this data seems to be also log transformed. Good. And if I look over here, I can see that, okay, still no sample annotations. So I'm going to use this data set to add some sample annotations so you see how that works. Before I do that, just mention the option that if you have a pre-normalized data set, which you can of course do, you save it as a text file and then you just load it via the wizard. So if you want to use DSEC2, for instance, you normalize it outside of the software and then bring it in via the wizard. Okay, now I want to add annotations to my dataset so I can analyze my groups of interest. And the easiest way to do that, I think, is to create the annotation file from the software. So I go File, Export, Sample Annotations. I give this file a name, that, save it. Then I open it, here it is, preferably in Excel. And here you see that what this does is that it takes the existing annotations, so the unique sample ID I have here. So if that's a very long sample ID, for instance, then it can be hard to type it in manually. So that's why I prefer to generate the file from the software, and then you can just add 1 column per annotation that you want to use. So maybe I want to have treatment and sex here. So here we have 2 control samples and 2 treated samples. And here we have a female, a male, a female, a male, like that. And we can add as many annotations as we want. And then we just save that, go back here, do File, Import Sample Annotations, find the file, okay, and then I have them here. So if I want to, let's just change the color there, like that. Now I can do a t-test, for instance, on treatment. See if I can find anything. Yeah, I'm looking at the Q value and you see that's not good at all. It indicates that most of what I've found could be a false discovery. But anyway, that's how you do it. And then, of course, after you've done this, you save this as a GE dataset. So the next time you open this, you will also have the annotations in there. Good. So, any questions on that? No? Then we continue. So now we will have a look at how you can download data from Gene Expression Omnibus. This is also a very nice feature to be able to access public data. So let's that. And then you go here, File, Download GeoDataset. And if you know the dataset you want to download, you just type in the number here and then press OK. That will connect with Geo, find the dataset, and download it like that. And here we were looking for a series matrix file. And if you are lucky, the series matrix file is there. So it depends on how the data has been uploaded. So more recent datasets are not always uploaded in that format. Then you need to do a more manual download of them. But older datasets are often available, and some of the newer ones as well. Anyway, here we are. We downloaded this dataset, and you see it comes with annotations. So I can start coloring here on the different disease subtypes and perform analysis on that directly. So this is very, very powerful like that. So in the case where you get an error message that there is no data available, which unfortunately happens because the data was not uploaded in the right format, you can always browse either the GDS, which is the older curated dataset, or the GSE, which are the newer datasets, and see, okay, so here it looks like some cell files are available. So let's open that dataset and see what we can find. So you can look in here, see if you see something. In this case, we need to open up a sample, and then we will actually find a cell file here, or a compressed cell file that you can download. So often when you go to the datasets, you can find other files to download, and then you can load them manually into the software. And there is also here, for instance, we talk about the SRA archive. So I need to open that. So process data are available on, raw data is available on SRA. And here were actually some other files that you can download. And we will do a webinar, I think, next week, where my colleague will talk about how you can access data from the SRA archive, if you're interested in that. So there's a separate webinar on that. Yep. Questions? No? Then we move on. Close down this. And now we will talk about single-cell RNA-seq analysis. So as you know, there is a lot of potential with single-cell experiments. That's why they are so popular. You can study the cell differentiation, which means that you can get higher precision in diagnosis of cancers and autoimmune diseases, also a better understanding of disease evolution and spreading. And when you are working with drug discovery, you can study the effect on different cell types. But it's also a challenge because this is quite noisy data compared to the bulk RNA-seq analysis that is also done. So noisier data, a lot of errors in there, and potentially very complex groups. But so in this example, we will use different techniques in the software to find potential clusters. We will use variance filtration or standard deviation filtration in the statistics windows to take away noise in the data set, take away variables with a low standard deviation. And then we will use clustering in PCA and t-SNE plots. And we will also use the k-means plus calculation to help us find groups. So this is a data set that has been made public by 10x Genomics and has about 2,700 cells from human peripheral blood. So let's have a look at that one. So I will go here, File, Open, and find the data set, which I have here. Here it is. So it's already saved as a GE dataset, so it has been pre-processed. And then you see that when data is stored as GE data file, we can just load it directly. So here we have 2,600 samples and 13,000 genes. And we see that, wow, here we need to do take away some noise in order to get this data set to open up There should be 2,600 samples in here, but it's just a very tight knot here. So let's use, so if we look here, it's not that easy to pre-filter. It's not obvious that you have a lot of zeros and so on, because this data is already log transformed. So let's instead use the variance tab here, where we can filter on standard deviation. So by dragging this slider here now, you see that I start filtering. Nothing has happened yet. Oops, that was too much. So we go back a bit. Yeah, I know where I want to go. So I type in 0.9. And that opens up the data set, as you can see. I now removed about 25%, and that opens up the data set, and it's possible now to see some different groups in here. And now I can either start annotating these groups from here, but I will actually move directly into a t-SNE plot. So I will change this to a 2D t-SNE plot. And the t-SNE plot is very popular, especially when working with single cell data, where the PCA plot can become quite crowded because there are so many samples. But the t-SNE plot is very good at clustering. So the t-SNE plot includes an active clustering. So samples that are believed to belong to the same group, they are moved closer together, and samples that are believed to belong to different groups, they are moved apart. So here you can see that we've found 3 very clear groups in here. And the perplexity parameter, that's your input here, and that's your guess on how big the clusters are. So you can test a number of different perplexities to see that you get a stable signal. And if you have a PCA plot side by side, you have quite a good view on how many clusters to find and so on. So you get basically the same result if I choose 50 or 100 here. So now I want to annotate these groups. So then I go to the Samples tab and create a new annotation, and I give that 3 more values, and I change that to a color that's easier to spot. And then I use the Annotate mouse tool, and I circle now this group here, like that. So that is now the blue group. And then I circle this little group. That is now the orange group. And then I circle this group, which is now the yellow group, like that. And there might be more groups in here. Here is maybe something, here is maybe something. But let's continue. So now what we want to do is try to understand what are these groups, these cell populations, what are they? So then we can use our knowledge about biomarkers. So for instance, I know that CD79 is a good marker for B cells. So by going here and just clicking here and then just quickly typing CD79, that will bring me to that one. So there are 2 of them. So let's color by CD79A first. And then we see that, okay, so we have a lot of upregulation for CD79A in this group. Interesting. Let's try CD79B, the same. So here we can guess that we have found B cells. So we can change, this was the yellow group, so if we now believe that, we can just change that to B cells, that. And then I have another marker for natural killer cells, which is, so I type that also very quickly, GNLY, this one. So if I now use that to color my samples, we see that we have a lot of upregulation here, which could indicate that we have natural killer cells here. Okay, so I could try to just circle this in the same way as I did before, but instead we will use another function in the software, which is the k-means clustering, which will help us to create the clusters. So let's take away everything else now and only keep the green samples. And so now we need to update the Disney plot again. So here we are. Okay. So now I can use this one here and say, I would like to create 2 clusters. I would like this algorithm to create 2 clusters. And these are the annotations we have so far. When I press run here, it creates something called k-means 2. So now I have an annotation called k-means 2 here. Like that. So this is the suggestion that it made. And well, if I'm not happy with that, I can continue and say maybe, okay, I think that there are maybe 3 clusters here. Then you can do that, run and see. Then you get the k-means 3 annotation. So that's a way for you not to be biased when you create. So maybe this was actually better matching what we saw before. So in this case, you don't create the annotation yourself, but you actually calculate the clusters using the k-means clustering. So that's another way of identifying subgroups. Okay, now we will move back here and activate everything again. And I will change back to the PCA plot and color according to this here. So one way to reduce the size of the data set, because if it's too big, it can be hard. There can be a lot of noise, as we saw, and it can also become very slow. So one way is to do what we did. We did a variance filtration to get rid of some noise. We can also reduce the size of the data set. So it's possible to create a subsample or do subsampling. And that means that I can say that I would like to create a data set that's half the size or maybe 10% of the size or something like that. And I want to stratify based on my new annotation. And stratification here means that I want the same proportion of samples in each group also in my new data set. And you can stratify based on 3 different sample annotations. Then I press OK. And that will create a new data set. Here we are. And so that's saved as a new data file. And given the name subsampled. And then we can see, so if I now color according to the new annotation and maybe filter down a little bit. See what we get here. Okay. Then you see that we have still the 4 clusters and the same proportion of samples in each of the clusters that we had originally. Yes, I think that was what I was going to show you today. So, questions? While you are thinking about that. Okay, so here is a question. How about just click opt when doing the filter by standard deviation? Yeah, you can try that, but it might not work so well with the single cell data actually. But you can definitely try it. But there is a very little variation on each of the axes when you have so many samples. So it might not give you a good result, but it's definitely a thing to test. Okay, is the subsampling done randomly? Does the subsample change by repeating? Yes, it does. It's a random process. So you can do it several times if you want to verify your results. Yeah, and we continue with webinars. Tomorrow is a very interesting one. As you might have noticed, the layout of the statistical dialogue has changed a little bit, so there are more options in there. And we also have something called Biomarker Workbench, which is a really nice functionality to give you a quick overview of a data set and also how the annotations are correlating to each other. And then I got another question here. Will this work as well for Aptamer-based assay data, SomaScan data? I'm not sure. I think it's not a platform that I recognize, so I think it's more like what type of files can you output from there? Can you output BAM files or text files? In that case, yes, we support it. If it's some other file formats that I haven't touched on here today, you might need to convert to the more general file formats. On Thursday, we will talk about the new genome browser, where we also now include support for ChIP-seq and ATAC-seq analysis. And next week, we have a couple of webinars. So we just continue to run these webinars and on different themes. So you will always be find something new and interesting if you're interested. And if you have any questions, just send me or my colleagues, if you're already in contact with them. Yeah, I see. I didn't put Jana in here, but if you're in contact with Jana in the US, you can also email her. Great. And then I got a comment here. In the SomaScan data, the signal expression are not whole numbers like RNA-seq data. Okay, but that's not a limitation here. It should work anyway. Okay, so thank you so much for your attention today. I will send out the slides to you, and I will also have a look at the questions that I got regarding the format of the gene length file and the allele balance. Yeah, see if I can find. I think the chat is saved, so I have the questions there. Great, thank you so much. Take care. Have a good rest of the day. Bye bye.